Welcome to part 2 of our lecture this week. So, back to this slide. You might ask yourself, if you have a DNA double helix, how in the world are we taking that and making new cells? So when I teach this for like general biology class or a life science class, non-majors type class, that kind of thing, essentially we think about eukaryotic cells having their plasma membrane, the cytoplasm, all the other organelles, and then this DNA is packaged in a nuclear membrane. So not only do you first start out with this chromatin, this really stringy DNA, but you also have these membranes in the way. So bacterial cells have a little leg up in this department because they don't have their DNA surrounded by a nuclear membrane. But even still, thinking about this DNA, the DNA is a double helix, so you might ask yourself, how do we take that and make more DNA? Well, it turns out that the DNA is very easily what we call unzipped. So remember G's with C's and T's with A's, but essentially, and there's a bunch of enzymes that are involved, but essentially the DNA double helix can untwist, okay? And it exposes those letters. So in DNA replication, we're essentially making a new strand from the old strand, and we're matching up the appropriate letters on the strand. So T's with A's with T's, G's with C's, C's with G's. Okay, so that's pretty nifty. Think about that process happening in the bacterial chromosome. So essentially, you've got your double-sided chromosome. Again, it's circular. So essentially, there's going to be a spot where that is going to open, and essentially, you're going to do the same thing. The two strands of the DNA are going to be opened, and then we're going to pair G's with C's and C's with G's, A's with T's, T's with A's. So now you essentially have this replication or making of another copy, right? This kind of reminds me of silly bands. Do you remember that? But when it's done, there's going to be what they call the nick, and that's where these two now copies of the original DNA are separated. So you have the one initial source of DNA, and then you have the two daughter molecules of DNA. So that's how that works. Okay, so now we're going to move on to a little bit more application. So you know that we have DNA. The DNA is part of the genome. You know, at least in statement, that DNA makes RNA. I need to describe how that works a little bit, and then RNA codes for proteins. In the classical biological literature, we refer to this as the central dogma. Your book refers to it as a central theme. But this idea that DNA codes for RNA and RNA codes for proteins, okay? DNA doesn't directly code for proteins, but DNA has the genes that can be then, we call it transcribed. The DNA is going to be used, the DNA of that gene is going to be used to form the first rudimentary information. We call that the messenger RNA, that then will be coded to become the proteins. Okay, so here on the right-hand side, a very simple, simplistic figure. DNA can replicate to make more DNA. Okay, much like I said earlier. Those nucleic bases are very easily pulled apart. You have enzymes that are helping opening up the DNA so we can expose those areas. G's with C's, C's with G's, A's with T's, T's with A's. And just like with the bacterial chromosome, then they replicate all of the DNA and then they separate. Okay, so not that much different in eukaryotic cells, but we call that mitosis. And there's a lot more involved. All right, so DNA copies through a process we call replication to make more DNA. Then we know DNA is going to be made up of genes and some other junk material. The genetic code for the gene then can be communicated into a strand of precursor protein, if you will. It's called the messenger RNA. We call this transcription. I don't know if you've ever heard the word transcribe. So I took a German class, actually about 4 or 5 German classes in college, and sometimes I was a bit lazy. Like instead of reading it and like already translating it right into a dialogue that made sense, I'd be like looking up all these different words, and I'd write down the words, and I wouldn't put the words in the right tense or if it was singular or plural, and then I had this kind of mess. And then I would try to like turn that into something that sounded like a conversation. So transcribing is getting that rough copy of the original German to English, for example. But it's so rough that it reads like, really bumpy, if you will. But translation is when you convert it into what would be more conversational, or if you're reading a book, it reads and flows really well. So transcription is when we take that DNA and we're going to make this rough copy, if you will, not really a copy, but a rough precursor to a protein, and we call it messenger RNA. Sometimes you see it just little m, capital R, capital N, capital A. All right. So both bacteria and eukaryotic cells have a cell structure called ribosomes, and it's on the ribosomes that we are converting that messenger RNA into proteins. Okay, so something good to remember, and I have a figure coming up next that'll help you remember that. All right, so translation is when we essentially then are going to take this messenger RNA and essentially convert it into protein. And there's actually another kind of RNA called transfer RNA. So there's a lot going on. I'm not going to dig real deep into this material on the exam, but just know DNA makes RNA, RNA makes proteins, and do know that that RNA is this sort of precursor strand to what will be a protein. But there's a lot going on. Also know that RNA is being translated into proteins at the ribosome. Okay, period. All right. Okay, so here's these words, transcription and translation. So if you want to pause and write those down, or if you're like, hey, I got a good understanding of this, then whatever. So let's take a look at exceptions to this pattern, the central dogma or central theme. DNA coding for RNA and RNA making protein. So you might remember in our viruses, we have DNA viruses and we have RNA viruses. So the DNA viruses, pretty straightforward, hijack our DNA to make more of its DNA. No big deal. RNA viruses are going to use RNA as a template to make other RNA. So that's a bit different. So RNA to RNA. And then we see there's the case of our retroviruses. So in the earlier lecture, we didn't really get too much into that, but now we can look at it. Take a look at the figure on the right-hand side. So if I have an RNA retrovirus, it is essentially going to hijack an enzyme called reverse transcriptase so that it takes its RNA and it makes DNA. And it's using the host genetic machinery. Then it takes the DNA and goes ahead and does transcription. So RNA retrovirus is taking the RNA, retroactively making a DNA, and then moving forward with making RNA and then making the protein. So a little back-asswards, but pretty novel if you ask me. All right, so let's look at some examples of how RNA retroviruses do their thing in situ or in a cell. So here is a retrovirus. You can see it has its spike proteins on it and inside the capsid, and then it has that genetic material. So you might remember that viruses are specific to certain kinds of host cells. So here we see receptors for those spikes. A spike is going to attach to the appropriate cell. Any cell that doesn't have the spikes is unaffected by that retrovirus. And then we see that the virus is taken up by the cell. You see endocytosis, that just means it's being engulfed inside the cell. Once inside the cell, we see we lost that membrane and the spikes, and so now we have the free capsid and DNA. I'm sorry, I meant to say RNA. So the RNA is released, and you can see here's this little piece of viral RNA. part of the information in that viral RNA is actually the instructions to make reverse transcriptase or code for reverse transcriptase. So that then is going to control the making of DNA from that viral RNA. And as we can see in our flowchart, then we're going to move forward with making the protein that's necessary for that, any of the proteins necessary for that RNA retrovirus, and eventually making a protein. So what we see here is this thing is copying itself through that process, and then assembling itself into new capsids, and then it's being released by that cell, then getting its spike proteins on the outside again and being released, as well as the envelope. Okay, so there is just some instructions there, or some information. The RNA of retroviruses is converted to DNA by the reverse transcriptase. As I said, that is coded by that original viral RNA, and then we move forward with our process. Okay, so some examples of retroviruses include HIV, and a lesser known, but it's definitely in the population, the human T lymphocyte virus. And then we have retrovirus of other animals, including feline leukemia, you might have heard that one, and SIV, which is another immunodeficiency virus, but in chimpanzees and gorillas. Now, we don't talk a lot about pharmacology in this class, but you will if you go on to nursing classes or if you decide to be a pharmacist or something. But essentially, this reverse transcriptase makes the treatment of retroviruses novel and unique, because we can use medications or drugs to target reverse transcriptase to stop the replication of retroviruses. So HIV really became in the media in the late 1980s, and essentially we know that it had a devastating effect on people who needed blood transfusions, people who, anyway, you know the story. I'm sure you know the story. But essentially, it adversely affected people who needed blood transfusions and individuals who, particularly male homosexuals. So the thing is so many people died from HIV. HIV, of course, turns into AIDS, and that's acquired immunodeficiency syndrome. So you don't have the ability to fight off any kinds of diseases. So it makes you very susceptible to even small things like yeast infections and pneumonia. And those infections can kill a person if you don't have the defenses to ward it off. So now we know, we've made leaps and bounds since the 80s. So we're looking at 35 years of research, and one of those areas of research involved using medications to target the reverse transcriptase so that we can kill the virus. All right, next topic is going to be about mutations. So when we think about mutations, we think about these as being changes in the DNA. It can happen in the DNA replication process, it can happen during transcription, and it can happen during translation. So we're really thinking more replication and transcription. But essentially, when we're dealing with genetic code and we're dealing with these letters, sometimes we may substitute a T for a C. And so that's how that happens. As eukaryotes, we have protective mechanisms or DNA repair systems that often will catch these mutations. Especially when we're younger, but as we get older and are exposed to environmental factors and so on, then we can have a higher rate of mutation. So let's think about bacteria. Not different than humans as far as mutations are permanent and heritable. So that means when you have it, then you can pass it on to your offspring. So most are not beneficial. In fact, they may have absolutely no effect at all, but some mutations can create variant strains that are more readily able to adapt to their surroundings, survive under new nutritional, situations or environmental situations, and may change their metabolism. And so maybe, in fact, they are given some special properties, and it may actually affect their reproduction. So it says, as long as the environment is stable, mutants only comprise a small percentage of the population, but we know that if the environment changes and that particular bacterium has some kind of advantage from this mutation that they may be more likely to survive.